All right, so everyone online, can you hear all right and see all right? I'm having a few issues with the displays, but I've got something on the board. Can you see what's on the board? Don't all speak at once.
Yeah, I can see what's on the board and hear you perfectly fine.
Oh, well, that's a good start, isn't it? Thanks, Cheese. Or whatever your name is. It's all right. What time we got? Oh, it's time. Wow, that's just in time, isn't it? Oh, well, it's interesting. A full classroom for once. Well, for the last 2 years, it's been pretty sparse, hasn't it? And those online, hello as well. And obviously, we're doing a hybrid thing here. So those online, if you've got a question, just post it, or you can actually just unmute yourself and speak up, and you should be fine. And those in the classroom, obviously, if you've got a question as well, you can speak up as well. So this is food microbiology. So if you're on course, it's a good time to leave now. And my name's Keith Warriner. I started here back in 2002, so it's my 20th anniversary. I'm not sure if we've improved since then, but we keep doing what we do, isn't it? So to start with, what I thought I'll do is I'll just go through the outline, which you can find on course link. And like I said, I'm having a trouble display, so I'll have to turn around every now and again. I don't know why I bother with this to tell you, but anyway, it's what it is. But you can see it yourself. So the first thing to note is there's 2 courses in here. There's a 3230 course and a 3240 course. So if you're in 3230, you do labs and you also do seminars. If you're in 3240, you don't do either lab or don't do seminars. So when I talk about seminars and lab, and you think, hang on a minute, have I missed something here? No, you haven't. Well, provided you're in 3240. So I'm going to go through 3230 first, and then we'll go through 3240, which is basically the same, except a little bit different. All right, so you go to the course link then, and we'll go to the course outline, and we'll go down a bit. It's going to be a hard job, this one. Anyway, oh, it's all right, keep going. Now, about this time, one of the people who does the IT comes in, but I bet he doesn't come in today. Anyway, we'll persevere. Anyway, so what this course does really is look at the microbiology of food safety with some spoilage. It's not like industrial microbiology, which some of you did in the winter. If you want to do industrial microbiology, you can sign up for that later on. So we're really focusing on what pathogens or microbes are associated with foods and what supports their growth. So that's the kind of main theme. We've put in sanitation, hazard, risk analysis, and all these kind of things into it. So as you can see, this is our lecture room. Means I have to walk up the hill. And worst of all, in the seminars, which used to be in food science, I've put them in here as well. So I'm going to get the steps in. So if you're in 3230, the seminars are held on Tuesday in McKinnon, which is, I think, well, it's here. And it's, I think it's the floor above being the number it is. And we'll talk about what we do in the seminar a bit later on. Now, if you're doing the labs, again, 3230, they are actually in food science. So in food science, if you go to the top, that's the one across the road if you lose your way. And if you go to the top floor, you can see the lab there, 202. And with the labs, you're divided into sections. There's some room in the front, so nobody likes sitting in the front. All right, fair enough. In the labs, there's sections. So section 1, 2, and 3, Monday, Wednesday, and Friday. There is some flexibility in terms of if you say, well, I don't feel like coming on Monday, so can I do the Wednesday lab? You could do that. But what I ask you, don't decide each week, saying, oh, what lab should I do today? Just choose your day and keep that for the whole semester. All right? Good, we'll talk about labs a bit. So in addition to me, we've got 3 dynamic TAs. Are any of the TAs online? There you go. So we've got 3 helpful TAs that you can contact about the labs. If you don't prefer contacting me, you can contact them, whichever you prefer. All right, so that we'll talk about that a little bit. So what we do, we've got a schedule. I try to keep to the schedule. I always fall behind, but you do your best you can, can't you? And then what we also have are guest lectures. So has anyone heard of Sylvain Chalbraz? Oh, he's going to be hurt. Sylvain is the one who's always on TV talking about Buttergate, dairy farming, lack of market. He comes on, well, I'll try and get him. He's quite a busy chap these days, but he likes talking, as you can imagine. So we'll have him. We'll also have someone called Jim Chan. So Jim Chan used to be the head of public health in Toronto. He's retired now, but his sort of fame, if you want to call it fame, was the outbreak at the X all those years ago, where 200 people got sick from eating at the food court. I'm pretty sure more people have got sick in the meantime, but he kind of solved it, or he got put up for it because he lost it. Oh, he was forced to retire afterwards, but we don't mention that. I know someone's got a chat. I'm just going to try and see what they want to know. Oh, that's all right. All right, so that's good. So yeah, we have guest lectures in, as I mentioned, and when the guest lectures come on, especially Jim Chan, and you want to go to public health as a career, here's the person to talk to, isn't he? All right, and obviously you can do a virtual lecture if you don't feel like getting up in the morning. You can just tune in if you really feel like it. I don't know why I put all those addresses there. All right, so again, 32:30, the lab schedule is, again, we do it the whole semester. We've got 8 labs in total. And, with these labs, really it's not to do with writing reports and doing things like that. It's getting the experience. So we're going to teach you how to do basic lab technique, and then how to analyze foods, how to do a sanitation monitoring exercise. We'll talk about how we do validation, microbial inactivation, thermal inactivation curves, a bit of mycology, and then at the end, we go over to public health if we're open. We haven't been for the last 2 years for obvious reasons, where we do food safety culture. So I don't know if that's something to look forward to, but that's what we do. And and I'll say it now, I'll probably repeat it. With the labs, what you do is that you don't really write a formal report, but you answer the questions in the course outline or the manual. And we'll go through the manual in a little bit, but what I try to emphasize is just make use of the techniques. Because I know when people are in labs and in groups, they'll sit back and say, look, you do all the work, I'm looking good here. Try to get as much hands-on experience as you can, because these are the techniques you need when you go to grad school, when you go to industry. All right, so the fun and games. So in the seminars, what we do is a presentation. Well, when we say we, I mean you. So what you do, you organize yourselves in groups of 4, and we're going to do a seminar next Tuesday. I'll give you tips on how to do a presentation. Just a few tippers and identifiers. So what you want to do is, again, 32:30, don't worry, 32:40, you don't have to do any presentations, is arrange yourselves in groups of 4. Now, obviously, some people are from different courses and don't know anyone here. And what we do is matchmaking on the Tuesday. So again, try arrange it beforehand. I just need the group names on Tuesday. You don't need to do a presentation. And if you haven't got a group, then obviously we can work on it in terms of trying to pair you up, things like that. Now, one of the questions is, like last year, for example, and the year before, to be honest, well, no, maybe last year, we did it virtually. And when we say virtual, it's a bit like this. Some people in the audience, some people online, some people recorded their presentation because they didn't like the live experience for some odd reason. And I'm not sure what to do. So I suppose it's a democracy, isn't it? So 32:30 people, if 32:40 people want to join in the can as well in voting, would you prefer a hybrid, or should we have it only in person? All right, fair enough. We'll sort it out on Tuesday. All right, here we go. So as I mentioned, what happens is that you do a presentation and range yourself in for, identify if you want to go first or not. There are advantages of going first. They're usually the people who get the highest score, but we'll talk about that in a little bit. Oh, no, we'll talk about it now. So in the group presentation, then you get into groups of 4. You've got to pick a subject on food safety or food microbiology. Like one year we had somebody talking about art for some odd reason. But anyway, keep it to food micro. Can be anything, food micro. So once you've chosen your subject, you write an abstract, and I've given you an example abstract on the course link. Then you give about a 25-minute presentation, and then you answer questions. Easy, isn't it? Now, the people who will be marking your presentation isn't me. It's basically the audience. So the audience will mark the presentation, and they'll upload their marks onto the peer review system, which you've probably all used before. And then what I do is mark the accuracy of the evaluation. So mark the marker, isn't it? So some people attempt to say, let's give everyone 10 out of 10 and things like that. But then if it's not so good, then obviously the person giving the 10 out of 10 will get a low mark themselves. And I'll talk a bit more about that on Tuesday, because the 3240 people are wondering what's going on about. But we'll talk about it on Tuesday. Is the best way, isn't it? All right then. So what we have then is 4 assignments throughout the semester, and these are all available now. So if you want to do them this afternoon, I wouldn't advise it, but if you wanted to do them this afternoon, you could do. And then we come to something called mind maps. So mind maps, well, I'll go back a little bit. So previously, what we used to do is ask people to write unit summaries, basically sum up what the material was in that particular unit. And I was talking to some students last year, and they said, well, we're used to mind maps, and it's quite interesting in that the different ways of learning, like in my good old days, as we call them, is that we used to learn things by writing. Literally the prop would be on the board with the chalk or acetate or this overhead projector, this museum piece there, and that's how we learned. Then I noticed back in the about 10s, I would call them, 2010s and things like that, when Google Images came along, people learned by pictures. And then I've noticed more recently, when I was asking for the unit summaries, people were all at sea saying, how can we summarize this information so in about 500 to 1,000 words? And I can see where the difficulty is now, because I guess when you could tell me, when you're at school, and when you were at high school, did you learn by mind mapping, or did you learn some other way? All right, fair enough. Anyway, so what we're going to try this year, I do listen to what people say sometimes. We're going to do mind mapping. Now, there's a lot said about mind mapping. As you rightly, like you probably got taught it one way, maybe several different ways. And I think with mind mapping, it's exactly what it says. Everyone has a different way of doing it to suit their learning style. And so this is one of the first units, as an example. And, the sort of tips you have is that you've got to try and make it compact. The fewer statements, use pictures where you need to, make summary statements, make connections between different aspects where you need to. It does go to a concept map then, but and it is difficult. If you haven't done it, is there any way we could do written summary instead? Well, no, because it's all consistent, isn't it? I must admit, I thought the summaries were right, but people were really struggling with them in terms of how to summarize the information. And in the end, some people were just copy and pasting the slides, which kind of went against it. So I think I can see the mind maps working quite well, actually, because they do give you summarized information, but how things connect. Has anyone got any other tips on how to do mind maps, apart from being concise, pictures, giving connections? You can stay online as well. You don't need to shy away from not. Nobody's got advice. All right, then have a go at it. Like I did this in half an hour, so you should be able to do it in less time. Well, hopefully. So the mind maps, I would suggest, oh bless you, I would suggest to do them for all the units because they're a good revision aid. But definitely you have to do them for units. Hang on, let me get back to my my spot again. I think it's oh yes, units 3, 4, and 8. So 3 is microbial growth, I think. 4 is sanitation, maybe, and 8 is no 8 sanitation. I think 4 is spoilage. Anyway, we'll work it out, won't we? All right. So we've got the mind maps done, and in terms of exams, it's going to be face-to-face exams. Last year we did write an exam. I thought it was quite interesting that, you know, basically last year, if you were in the industrial micro, you can rewrite lectures as well, where the exam was for you to write the exam and then submit it to me. And it's amazing how brutal the questions were on those exams. And if I actually did them, I probably would guess, hang on a minute, this is too hard. But I thought it was an interesting exercise because it's harder to write exams and mark them than to actually do them. We don't appreciate that at the time. But what we do is have a midterm, and the midterm's composed of short answer questions, multiple choice, and one long answer. The final is the same, obviously covering more material, but you know, let's look forward to that when we look forward to it. All right, so again, the laboratory reports, we'll talk a bit more on Tuesday about these because like 3240 people will be getting confused. So you go to the lab manual, which is here, and you just answer the questions after each lab. But anyway, you know how to do that. All right, so the last thing I want to talk about 3230, which again is like 3230, is the quizzes. So what you'll notice is quizzes on the quiz tab. You can take them at any time. Obviously, the best time to take them is just after we've covered them in the unit. And what these quizzes are really about is just assessing if you've got the knowledge. Because when I talk and give her all this information, you say, well, what should I remember? What's the important part? And I think what the quizzes do, if you can answer the quizzes, then you're in a good spot. You know, obviously, you know the material. You do get 4 chances. So even by the laws of probability, you're going to get 100%. But I would just, like, you've got till the end of the semester, but don't wait till the 2nd of December and say, I'm going to do the quizzes now. I would suggest to do them after each unit, but it's up to you, as always. All right. All right, yeah. So we don't have a textbook for this course. The reason why we haven't got a textbook for this course is because no one textbook covers all what we need. You know, because when you look at the fundamentals of micro, that's basically just micro, and a bit of gives you a list of names, and that doesn't really touch HACCP or food safety systems. And likewise, those that talk about HACCP, risk analysis, don't talk about pathogens and that. So when I retire, if I get that far, you know, I'll probably write a textbook, maybe. I probably won't. Anyway, it's always good intention. So what we do then is not mandatory of a textbook. But if you want to read a textbook, to read out the area, to make preparation for your presentation, to answer assignments, then there's on course reserve, there's these 2 books here, which you can refer to. But as I say, you don't need to buy them. There's no field trips, unfortunately. Go to Industrial Micro if you want that. So with the 3230 people, and is there any questions on anything? Oh, it's going very quiet semester, that's good. All right, so we'll go to 3240 now. So with 3240 people, which I think is half the class, to be honest, more than half, it's basically the same without the labs and the seminars. What I forgot to mention is that on the front of the course link, there's an announcements page. And so we post things of interest. So I got this last night from our friends in the Innovative Biology, the ones with all the money. And I've got this mentorship program. I don't know anything about it. I don't know what's the purpose or whatnot. Well, it's something to do with applying to graduate school, I suppose. But we post things like that. And I noticed the TAs posted something last night as well. They didn't. But the only difference is I'll just go through it. I won't go through it all, obviously, it's more or less the same. Is the oh, where is it? You'll go to Dropbox. Yeah, so I forgot to mention all the assignments and everything I put in Dropbox. So for the mind map, for example, you might say, oh, it's too tedious to write in PowerPoint. So there's nothing wrong with doing drawing, taking a picture, and put it up to the Dropbox. But everything has to go through the Dropbox, because I don't just email it, because I'll think I'll lose it. And I guess the TAs might lose it as well. But no, so everything goes into the sort of Dropbox. And they've got these dates, and these dates might change depending where we are. Yeah, we usually stick to, well, we do our best to stick to them. Now, the big difference with 3240, apart from no lectures, apart from no seminars, is the hot topic. So this is a term paper, about 1,500 words, where they pick a subject in microbiology. It could be the romaine lettuce outbreaks going on at the moment with Wendy's, or it could be what's the other outbreak? Yeah, it could be about listeria and cheese. There's one going on now. Whatever it is. So with this term paper, you just pick the subject, you talk about its importance, you give the background information saying, oh, this outbreak happened here, this is how E. coli gets into lettuce and whatnot. And at the end of it, you give an opinion of what should happen or what should be the next steps. But just read it. Like say, if you get, it's not due to the end of the semester, but don't leave it till the 2nd of December, hoping that, oh yeah, I can do that. So that's the difference there. Again, you've got the grades list, which you know, we'll talk about that, and the coursing help and the quizzes as per use. So is there any questions from the 3240 people about that? Oh. Not really talkative, but anyway, it's all right. Right. So, all right then. So, have a read of it, you know, and then we'll sort it out after that. So, what I'm going to try and do now is go to the PowerPoint, and if it's going to be as easy as I thought it was going to be. We get better for this technology, don't you? So I've got you. I was going to say, have we got a doctor in the house in terms of have we got somebody who knows how to work these things in the house, isn't it? Oh. Hang on. So I've got it. Yeah, like I say, all these are messed up a bit. Oh wow! Well, it's pure fluke that. I'll give you that. All right then. So, can you see the slides online? All right. Is anyone awake still, or? Yeah, like I say, last year I was doing this.
We can.
That's good. Last year I was doing this, and it wasn't on the. They couldn't see the thing, even though it was posted up there. But they never said anything, which is good. All right then. So, just to confirm what I've just said, I think I'm boring you now. Very early on, is that we do the lectures in here 8:30 till 9:20. We do go live, as you can see, and I will post the recordings on course links. So if you're in 3230, you go to the Zoom tab, go to recorded lectures, and it'll give you all of there. For the 3240 people, I basically put them in a folder on the course link which says recording, so you can see them at your leisure. So 3230, as I said, you've got your labs, which is in the top floor. We have different groups, the sections, but if you wanted to move section, you can, but once you've moved, you can't move back, so to speak. The first lab will be next on Monday. So we start off early, and it's just basic techniques for the first lab. For the seminars, 3230, we have to go upstairs, I assume, have to find that lecture room, and that's for giving presentations. And in terms of the breakdown of marks, obviously for the assignments, they're going to be fairly easy, not big assignments. You should take you about half, well, maybe an hour or two. And they're worth 2.25%. Your units or mind maps, as I keep calling unit summaries, 6% in total, and the midterm and the exams and the quizzes. And the 3240, as I said, it's exactly the same, except no labs, no seminar, and you've got a hot topic discussion paper at the end. All right, so is there any questions? Oh, I will sleep here, yawning over it, that's not a good sign. Is there any questions anyone has? Fair enough. All right then, so before we get on to the subject of, well, it is the subject of food microbiology, I think one of the things we have to appreciate is our sort of viewpoints on microbes, because everyone thinks, well, they're very simplistic, single cell usually, dead ends in evolution. But when you think about how things evolved, you'll notice how complex it is. And there's no such thing as a single cell per se, because a lot cooperate together. And this cooperation, this evolution into specific niches is valuable information to the food microbiologists, because they want to know what conditions would these pathogens grow, for example, to cause illness. What conditions do we need for them in order to get them to grow in the lab? And a bit like spoilage microbes, what makes them grow and spoil products, and how can we stop that growth? And we only know that by knowing how they've evolved in order to become associated and grow. So what we do first is that we think about evolution and think about how we do things and how things arose. So I don't know what you learned in high school, to be honest, or whatever, but there's different theories about how life evolved on Earth. One theory is the tidal flat theory. I'll just try and highlight it. Ooh. So the tidal flat theory is where you've got this primordial soup of all these nasty sulfur, iron, ammonia, things like that. And on these tide flats, you've got the perfect conditions. It goes dry, it goes cold, it goes wet, it goes pH goes acidic, goes to alkali. And this unique chemistry actually brought about the building blocks of life, which then came together and started to multiply and grow. So that's one theory, the tidal flat theory. The second theory, which is interesting, I like this sort of the panosporium or sporium. And what this theory is that life was seeded on Earth. So basically you had meteors and comets, meteors, isn't it? With the building blocks of life already. And they landed on Earth and made a seed like you would grow a plant. And life, they just like bloomed from this. Now, Neo-Panisperia, which is this one, and the spermia, I keep getting that wrong. That's where this is an interesting theory in that, whereas Panisperia is to do with seeding life on Earth, Neo-Panisperia is where it's constantly being bombarded by microbes. So the theory goes is that we're constantly getting new microbes showered on the Earth. And people thought, yeah, this is crazy, isn't it? But when they actually started looking at microbes in the stratosphere, they're interesting, as in they're not mole spores, there's viruses, there's bacteria, but there's no moles, no pollen. Because the natural theory, isn't it, is that, oh yeah, well, those in the air, come from the ground. But so what they're suggesting is that no, raining down on us, and those that survive, basically proliferate. You know, one could even give a theory about the coronavirus coming from outer space. So why not? The theory is as valid as what they've got now. But it's an interesting theory in that on Venus, for example, is suggesting that microbes live in the air on Venus. Maybe it's crackpot, who knows? But anyway, interesting theory though, isn't it? Then we have we're all Martians. So basically what happens is, meteors from comets, no meteors, isn't it? Came from Mars, and Mars was inhabited at one point, and the waters were flowing, and for some odd reason, the sort of microbial life descended on Earth, and we all kind of adapted and grew, and we're all Martians looking for Martians. There you go. That's ironic. The other theory is that we have, I'm trying to look at number 4, I can't even see it. Oh yeah, the oceanic ridge. So basically what this theory, I guess this is the more sort of common theory, is that, so you got these sort of hot springs and they produce sulfur, ammonia, and all these kind of things. And the conditions were right in order to build these building blocks of life, the nucleic acids, the DNA, the protein. Now, a similar theory to that is the nuclear geyser and I guess the volcano. So what it's suggesting in these two theories is that we needed something to react to actually produce these building blocks, the proteins and the nucleic acids. And because the primordial soup was this sort of toxic sea of sulfur and ammonia, acid, probably strong alkali, there was this sort of golden spot where life could adapt and then spread out as the Earth kind of became a bit more livable. So there's different theories on. There's no way of proving any of them, obviously, you know. And if you did come up with a theory, they'll probably call you a crackpot. But we know that life did because we're here to say it. But why do you think molecules had this urge to multiply, replicate? What was it that drove it? You can answer online as well. Oh, I thought we were going to get some earth-shattering thing of what the purpose of life was, but I think we'll have to wait for another year, isn't it? Yeah, it's quite interesting in that, it's very philosophical, is that say, all right, you got these molecules, but what made them the urge to grow? What made them the urge to evolve into the complex things we are? What is the ultimate goal? But again, we'll leave that to the philosophers, should we? All right, then. So basically what happened then is that we got the building blocks. And when we talk about building blocks, we talk about nucleic acids and proteins and hydrocarbons and things like that. But that's only one thing, isn't it? Like you could have a tube of cysteine, but that doesn't mean, say, if you wait long enough, it's going to evolve into a life form. Things have to happen. And with life, isn't it? It's almost like you're passing on instructions to make a new you. So when we get new offspring, they're passing our genes on. When bacteria grow, they pass their genes on. And really, you can boil down life, isn't it, into passing on genes. I suppose that's the best way of looking at it. But what people are questioning is, what was the initial blueprint? What was the initial blueprint to make these replicating cells? Was it RNA? Well, certainly RNA was formed, later on DNA. And RNA is an interesting sort of molecule, if you like, because not only does it have genetic information, but it can turn up on itself to make interesting shapes. And these shapes can be receptors, it can be enzymes, up to enzymes, isn't it? And proteins, on the other hand, well, yeah, they kind of transfer information. We've got prions, which we'll talk about at the end unit, where these kind of replicate in a way in that they make good proteins into bad proteins, but it's not that simple. But that's a kind of replication. It's making copies of itself, even though it's not making copies, it's converting pre-existing proteins. And obviously they're catalytic. They actually can do the business in terms of making these reactions happen rather than having nuclear hot springs all the time. So what came first, do you think? Was it RNA or was it proteins? Oh, go on, please. Yeah, I think I'm actually talking to myself here. Am I in the real world? No, good. It's a good theory. Nobody knows. So it could possibly be, but what the sort of theories, and these you could literally come up with anything. It's a bit like the if you're an astronomer, you can say, oh yeah, this is a black hole. This is the oldest thing going. Who's going to prove it? But basically, you're right in that these proteins, nucleic acid, develop together, and this likelihood is what happened is that there was this sort of mixing, transferring. And what people suggest, and like I say, it could be anything, is it's the vesicle, the lipid self-assembly membranes, that's actually the key to life. It's not to do with nucleic acids, although they're part of it. So with vesicles, these lipid membranes enable the chemistry to be contained within, rather than being floating around, contained within this thing. And if you go to a TED talk, which was done in 2014, I think it was, there was this sort of presentation on the responsive oil droplet. So what these people did, I forget what university they're in, is they made these oil droplets out of different lipids, about 3 different lipids, put them together in a self-assembly. And when they put them in solutions and did a pH gradient, the droplet would swim towards it. When they put sugar in, the droplet would migrate towards it. When they put proteins and nucleic acids in these droplets, they found they started to fuse and then spread out and things like this. So what their proposition was saying, yeah, this is how life formed. It was the vesicles, the chemistry inside, and for some reason they decided to replicate. Now, as time went on, obviously this fission was random. You got random proteins, random nucleic acids, but something happened where they said, well, this is very inefficient. Let's try and get some copying of a specific genes. And so was born the viruses. So what these viruses were doing is taking the sequencing of these genes and then going to different vesicles and basically multiplying as viruses do. So basically viruses were copying themselves, but eventually what was happening is that you get mismatches, you get mutations. And so rather than just copying themselves, we started to get this evolution of the cells we know today, because they've got enzymes, they've got metabolic pathways and things like that. And so these viruses are with us today, obviously, because we're living with one at the moment. And that was the seeding of life and the sort of dynamic of replicating clones, i.e. the clone, isn't it? And we got this divergence as the genetic material mutated, new genes were acquired. So all in all, the theory of life, and like I say, you could come up with your own theory and nobody will know when you can't say any different, is this sort of parallel evolution of the lipid vesicle, the protein and nucleic acid. And it was the conditions at the early Earth, the extreme in pH, the heat, the cold, all these things coming together to make this vesicle, which could then replicate by fusion. Yeah, it's all right though, isn't it? Like that could keep going for a few million years. But really, what the big step was is the evolution of metabolic pathways. So you can see yourself living at a vent, and it's very competitive. You can't stray away from the vent because all your sulfur, your energy sources are at the vent. But you think, well, no, let's spread our wings and go away. So we started getting evolution of what we call the bacterial cells we know today. And to become independent, you basically had to come up with stuff. You basically had to come up with your own powerhouse, your metabolic pathways. And if you do industrial micro, we actually talk about how metabolic pathways evolved. An interesting thing, but I'm digressing if I don't. So what you also had to do was increase your stress resistance, because outside of the happy home of the spring, you're going to be suffering from acid, heat, cold, all these things. And you basically evolved. And long story short, the early bacteria, you probably learned in your micro, which you've done before, is the Archaea bacteria. these sort of primitive bacteria, which have got not peptidoglycan, but pseudomurine as a cell wall. And what these were, the extremophiles. And this is why people think, saying, well, the first bacteria were the ones that are tough, could survive in the spring, with the thermal, the high salt, methane, you name it. But obviously they evolved into eukaryotes, which eventually were the pinnacle of evolution, I suppose, and the bacteria. Now, with the bacteria, as we mentioned, when they moved away from their sort of vesicle form, they actually started developing cell walls. And cell walls are critical. They give you rigidity, they give you protection of certain kinds. And when we look at the bacterial kingdom, we see two types. We see the gram-negative and gram-positive. So the gram-positive has a very thick peptidoglycan layer, but no outer cytoplasmic membrane, whereas the gram-negatives have an outer cytoplasmic membrane to give protection, which has got something called LPS, lipopolysaccharide. And this LPS in pathogens is really significant. This is what our body detects in our immune system. It's hydrophobic, so it kind of pushes away anything that's hydrophilic, but it's got a very thin peptidoglycan. So what do you think came first? Do you think it was the gram-positive bacteria or the gram-negative? Yeah, that's right, gram-positive, isn't it? Because basically gram-positive, oh, bless you, gram-positives are resistant to desiccation and heat, broadly. So that's what you need. But when you get out into the real world and you've got all these microbes trying to beat you up, you want this sort of membrane protection. Again, there's different theories. Some people think gram-negatives came first. Some people think gram-positives, and there's no way of knowing. But basically, you'll see how significant they are in terms of identifying them, and also the difference in resistance and how gram-positives and gram-negatives react. Now, the gram stain, which was developed by Hans Christian back in 1860, I think, could be wrong, that differentiates the bacteria. And we all know what the gram stain is. The gram stain is where you put crystal violet iodine to form a complex, then you add the ethanol, which would then strip the outer cytoplasmic membrane, and therefore you'll get, then you counterstain with saffron. So what you'll get is the gram negatives appearing pink because they've been stained with saffron, whereas the gram positives have the iodine crystal violet complex. Now, as we talked about with gram stain or gram reaction, you'll find the membrane protection is very significant when it comes to sanitizers, antibiotics. Whereas the gram-negative positives, they tend to be more stable. They'll literally live on the surface. They'll be thermally resistant. There'll be desiccation resistance and things like that. Now, not all bacteria in a population has a cell wall, and those which lack cell walls are called L forms. So any sort of microbe, bacteria, should I say, gram-positive, gram-negative, doesn't matter. They'll have a small population called L-form bacteria, with no cell membrane or cell wall. And some people are like, say, oh, it's just a relic. Basically, this is just a relic from the past because they don't do binary fission. They act more like the vesicles. They kind of be those lava lamps used to have in the 70s, where they'll break off, rather than binary fission, as we know with bacteria. But the reason why people think they're significant is because of lack of cell wall, they've actually got less targets. So if you're antibiotics, for example, they mainly target the cell wall, and that's how it kills bacteria. But if you've got no cell wall, you can't be inhibited. They're very firmly resistant. And what was interesting is that people think they're also associated with latent infection. So when people look for L-form bacteria, to find them in lungs, for example, just waiting for their time to shine in terms of going under outgrowth. They're very slow growing. They need hypertonic, i.e. very high salt concentration to stabilize them, because the cell wall's no longer there to stabilize them and they'll burst. So L-form bacteria, some people say, oh, it's just a lab curiosity. It doesn't happen in the real world. But as I said, people are actually finding them in the real world now. So really what we're saying is that the development of cell walls, especially for the bacteria, really were some of the key parts or the key step in evolution, because they provided the rigidity, the physical protection. So they could now start living outside the vent. They've got the metabolic pathways, they've got the cell walls. And as we go through the course, we'll talk about stress resistance of different microbes and how they cope with it. And when you look at how they cope with it, you'll look back at evolution, say, yeah, that's what they needed to survive between hosts and things like that. And with the cell wall, not only did you get the rigidity, but you started with the genome. And what by that we mean that when we just got no cell wall and just vesicles, like viruses or DNA was coming in and going into the vesicle, was no continuation. Because evolution is basically the continuation of the gene. It's not to do with viruses, it's not to do with bacteria, it's not to do with eukaryotics. It's basically transferring genes. To what end, who knows, but there must be some kind of end, isn't there? So it gave you that stability, and it enabled them to diversify because of that stability. You've got genes, you've got genetic lineages like family trees and things like that. All right, so that's the crash course in evolution. Interesting area, like I said, when you look at what people think and their sort of theories, but that's life. So, as I say, we're in a position where we think, yeah, we've got evolution sorted out, which we haven't. You know, basically, we've got as much clue as the people back in the 400 BC have. But with Aristotle, for example, who was a famous Greek, I think, philosopher, could be wrong, could be Roman. No, I think he's Greek. Obviously we have to explain evolution. You know, what is it? How did it happen? You know, why did it happen? So back in the day, Aristotle said, well, really it's to do with, we got all these different creatures, but it's all spontaneous. It's all very random. And those that survive in the environment will go on to grow. Those that aren't adapted die off. And in a way, he was quite correct, wasn't he? But when you think about evolution right back at the start, is that, yeah, those that could survive and flourish did, so those that couldn't died off. So he was more bang on, maybe, I don't know. So, you know, obviously after the fall of the Roman and Greek empires, we went to the Dark Ages, which nothing really got done, apart from a few good Netflix films. We got to the point of Darwinism. So with Darwinism, you know, he went to Madagascar, I think it was, saw the life there was very different from this sort of land, from what he's used to on the land, this island of Madagascar. And he came up with a natural selection. So a bit like Aristotle, he said, it's not so much random survival, it's basically the environment which shapes the of the life that can flourish and go onwards. Very much like, he was talking about big animals, not bacteria, but again, it's like bacteria. You know, you get a lot of bacteria coming in, but it's only those that survive and flourish that become dominant. And so this natural selection, survival of the fittest, you know, it's the individual that's adapted. If you've got a longer neck and can reach up to the trees, you're going to do much better than those with short necks who can't reach up to the trees. And basically that's what it was. You know, I guess it was right in certain respects, as we talk about. But then we got this built on the theory saying, well, you know, life in evolution always goes to complexity. You know, basically, it's like a tree growing. It has to get more complex, and we're at the top of the tree and things like that. But, you know, in reality, what people are thinking now, there's Darwinism evolution, for sure, survival of the fittest, but then there's the reductive evolution. And what I'll talk probably a little bit about is what we call Hamilton evolution, or cooperative. So we have not only one type of evolution, which is the survival of the fittest, we also have this reductive and cooperative. So, yeah, it's 9:19 now. I won't launch into our friend Hamilton. Have a look at the course outline then, and get back if you've got any questions. The 3230 people, yes, there is a lab for group 1 on Monday. Yes, there is a seminar next week. In the meantime, 3230 people, get yourselves into groups of 4. You just don't need to give me a title, just give me your names and if you want to go first or not. And then we'll start up again on Monday. Oh yeah, so yeah, found a prion that had cooperative function. Yeah, like I say, it's an interesting sort of notion when we get to prions in that it's not so much conversion, but it's more to do with, well, it's the conversion rather than going on. But I think there's definitely more than one way of replicating. I remember there was a paper just last year where we were talking about inorganic replication, this crystal that used to replicate itself. And you think, well, yeah, it's life, isn't it? But anyway, good question. That's all right.
About my bat